And so this session is on applications and timescales, and Desiree Tomazi is going to start.
Good afternoon, everybody. It's great to be here to start off this session. And so we're going to start looking at some applications of forecast information at different timescales to support fisheries management in a changing ocean. So I'm Desiree Tomazi. I'm at the NOAA Southwest Fisheries Science Center, also affiliated with the University of California, Santa Cruz. And I just want to acknowledge my many collaborators that you'll see their names in the slides and references to their papers. So I just want to make sure that you go on and check out those references that I'm going to be speaking of. So Mike put up these figures, but I just want to stress that fisheries decisions across timescales are impacted by climate effects. And that is because we know climate affects vital processes of living marine resources as well as their distribution. And then this leads to these processes that I've highlighted here in the clouds, the climate processes, affecting a lot of the decisions that are being taken. And those decisions are taken at a variety of scales. And I've highlighted some of those in those colorful cloud blobs there, as well as a more specific one on the side. Oh, now it's not moving. Okay. And in talking to stakeholders, and actually I looked at a poll that was taken of stakeholders on the West Coast that are engaged with the Fisheries Management Council process in advisory bodies. And it was clear that there is a need for climate-ready, what we call climate-ready fisheries management, or information that might give you a foresight of what species of interest to the Council will be doing 1 month from now to on the century scale. And those relate to decisions that are taken during the fishing season. So we call those in-season fishing decisions to the famous stock assessment or setting the catch limits, and that's usually taken at an annual or a biannual scale. And then there are also longer-term strategic issues of do we need to change our current management or harvest control rule because it's not robust to projected changes in species vital rates or distribution under climate change. And so you use different models and different approaches when you're looking at these sort of different decisions. And today for this talk, I'm going to really be focusing on sort of the yellow and green boxes and not so much on the strategic decisions and the century scale time frame. So going back to this first slide, I'm going to present application from this pink blob all the way to the green one. And I kind of color-coded my slides so you can sort of follow along in terms of the time scale. So I'm not going to spend too much time here because the speakers after me can talk way more eloquently than I can on this. But I just wanted to let you know that outside of NOAA Fisheries, the NOAA National Ocean Service actually have a NOAA Ecological Forecasting group, and they're looking at forecasts on the near real time to really the short time scale, so up to 5 days outlooks for some of these. And these are related to harmful algal blooms, coastal pathogen forecast, hypoxia forecast, and coral bleaching forecast. And I'm not going to talk about this today, but in terms of coral bleaching, they're also they have developed experimental seasonal forecasts for those. On the fishery side, we have a few near real-time or nowcast, like the EcoCast tool that Mike was speaking to this morning. And so these are dynamic ocean management tools that are developed to reduce bycatch. So you have nowcast of ocean conditions, and initially those were taken from actually satellites. Now they're also taken from regional ocean models, but then are linked to species distribution models. And these species distribution models are often they are statistical models linking the physics to the distribution of a species of interest. And they are often developed using machine learning methods. And then in this case, EcoCast uses species distribution model of a target species, like swordfish, as well as a few of the bycatch species that are of interest to the council because they want to protect these resources. And these are loggerhead turtles, sea lions, and also they're including sharks. And so you see here on the left, and then this product is freely available on the web, and fishers can go look at the map when they're deciding where to fish, and they're saying, okay, maybe I should avoid these red areas because there is an increased risk of bycatch. Now, nobody has actually sort of kind of followed up to see is this actually being used or not, but they're definitely tracking the amount of bycatch in this fishery. And so that's work that's done by Elliot Eisen at the ERD lab, in the ERD group in the Southwest Fisheries Science Center. And that same group has also developed Whale Watch, which is similar, but it's looking at producing maps of whale distribution. And for the current month, in this case, so you can see the scale over here, but now they're updating it, if you look at the Abrams et al. Paper, to being a daily nowcast of whale distribution on a 10 by 10 kilometer scale. And again, this is to reduce human impacts on whale, in particular ship strikes, but it could be useful also to reduce whale entanglements with fishing gear. And then out in Hawaii, they have another similar product. It's called Turtle Watch. And this is simpler in terms of the variables, physical variables that are ingested into this product. It's really looking at the preferred sort of thermal habitat of sea turtles. And so again, this one ingests satellite information, or really just SST, and produces this map. And you see this area kind of enveloping 63 Fahrenheit up to 65.5 Fahrenheit. That shows the preferred habitat for the sea turtles. And then again, these are sport fishing vessel, a note to avoid longliners, avoid those areas because of increased bycatch risk. Then another way that the species distribution model can be used and are actually being used is to optimize survey coverage. This is actually happening on the West Coast. So we have 2 cruises going out every year that are the coastal pelagic surveys, and especially the one in spring is aimed at sampling the sardine spawning habitat. And so Juan Zwolinski and Dave Dimmer at the Southwest Fisheries Science Center have developed a species distribution, a spawning habitat model for sardine. And every time the crews go out in real time, they look at the map of the potential habitat to ensure that they sample the entire habitat. And so I wanted to also, give you guys a list of the environmental inputs that are going into the species distribution model. So as I said, some can be very simple, like the sea turtle watch product, and just include sea surface temperatures, but they often include, in the case of EcoCast, other variables, and like sea surface height, eddy kinetic energy, something about the surface winds, and chlorophyll A is often very important, and even subsurface variables like isothermal layer depth or the ground physalia frequency. So now we are moving to a different time scale and looking at the seasonal time scale. And so some of these approaches in terms of the biological models, so the species distribution models, have also been used on the seasonal scale. And way back in 2011, Alistair Update, that really pioneered this approach using seasonal forecast for fisheries applications, has this product. This is now in Australia. We are changing continents, where it's linking a seasonal prediction system. So this is a dynamical seasonal prediction system, and taking just SST from that, SST anomalies, and then adding those to a climatology. Because I guess as we were discussing before, they've done the habitat model really ingests the full temperature, not the anomalies in the ways that they're constructed. And then that informs a southern bluefin tuna habitat boundary forecast that is stable for up to a 4-month lead time. And that leads to improved operational decisions in terms of the fishermen. They can take a look at the forecast and say, okay, maybe my effort should be spent fishing further north because it seems that this year the zone of kind of the buffer zone and then the core zone is moving northward. So I'm going to have to fish even further north. And on the west coast, Stephanie Brody at ERD has looked at sort of potential predictive scale. So this is kind of an idealized model in the sense that she was not using a forecast but sort of perfect information in terms of the physics. And she looked at, what's the predictive scale of swordfish catch anomalies. So if you remember, swordfish was the model that was part of EcoCast. And an thing that she did is that when she was looking at a summarizing prediction scale, she looked at it at different resolution from 0.5 degrees all the way to the entire core fishing area for swordfish. And she also decomposed the environmental signal that went into the species distribution model from just the climatology, which is the purple line, the yellow line, which is the climatology plus the low frequency signal, and then adding on top of that the daily fields. And here we are looking at the upper tercile accuracy, so more of a probabilistic metric, and the dotted line is just your random forecast. You can see that maybe for some months there might be some scale, above just a random forecast. So there is some suggestion that maybe there is potential for a skillful seasonal forecast of swordfish. And I guess what she found was that the low frequency component was the dominant source of predictive skill. So you can see that adding the daily field doesn't add much. And you can also see that if you're averaging over a larger area, then your skill, you can see here, is increased in some months. And the environmental inputs here were SST, chlorophyll A, and ILD. And then again on the west coast, and Sam will speak more to the J-scope downscaling model, but they've actually produced a hake distribution forecast. And so I think there is a global seasonal forecast system that feeds into the J-scope RAMS model that downscales it. And then this produces the temperature at the depth that it's important for this fish species. So this is 8, so it's not surface temperature, but at 250 meters deep. And that feeds into the habitat model. And he looked at the scale of an 8-month lead time forecast. They didn't have a lot of years to do out of sample validation of this forecast, but it seemed promising. And so they're providing these maps on the J-scope website. And the hope is that this would improve survey planning, sort of like the sardine, but at the seasonal time scale. And also could give an early warning of ecological shifts because the hake is a prime, it's a predator on the west coast. And so it would be interesting to know of changes in their distributions for the managers. And there's not a lot of work that has been done at the multi-annual forecast scale, especially in the US. But in terms of stakeholders, I think that would be useful for survey planning, also avoiding international conflicts over fishing rights. If you're thinking about distribution, at dinner I can talk to you all about the mackerel wars in Europe. And also, obviously, helping the stakeholder plan for changing fishing opportunities or for changing interactions. And on this one is work that has been done in Europe, where they produced multi-annual forecast of suitable blue whiting spawning habitat. And they're actually a big driver. They tried both salinity and SST, but really the driver in terms of the predictability was salinity. And that's because the low frequency dynamics of the North Atlantic subpolar gyres give quite a long term, are a good source of predictive skill at this multi-annual scale, and they produce the. So this is a metric of forecast scale, and just the three colors here. The blue is a model of blue whiting habitat, where the input in terms of salinity comes from a dynamical model, and then the other two are sort of persistent products, just different persistent products. And you can see that at least for the 1 year to 2 year scale, there is some additional scale brought up on by the dynamical model, but persistence does pretty well in this case. And this is the scale of the biological forecast, just driven by these three different products. And now we're going to go to forecast of stock abundance of biomass. So we've been talking a lot about distribution, but what stock assessment folks do a lot of is producing these forecasts, 1 year out, maybe 3 year out, of what the stock biomass will be, and that is the basis of then their catch limits. For some species, like North Pacific tunas, you actually do 10-year projection into the future, and that also informs catch advice. And just a little bit about all the potential sources of maybe predictability, well, in those, just from the biological side of things. But initial conditions, you can think of this as biological persistence, are very important to giving you some scale in this forecast. That's because how many fish you have today is going to be an important determinant of how many fish you will have next year. So the age structure and the biomass at age, and these are all done with population dynamics models that really take advantage of the age structure of the stock. And then obviously productivity also matter. And so what recruitment is going to be, mortality, as well as growth. But then also fishing. So if the stock is fished really hard, fishing will obviously determine the biomass next year. And so that's also related to selectivity. And actually, I wanted to stress that fishery scientists call these projections, because it really depends on the fishing mortality scenario that you're using, and they often compare different mortality scenarios to see, okay, given this condition, if we use this fishing mortality, what's going to happen to the stock, what's going to happen if we use another one, and then they could pick an optimal one. And so here, just thinking about how the life history of each species is going to contribute to this biological persistence. So here is the lead time in years, and this is something that, like a conceptual plot that we put together. But shortfin mako shark, for instance, has a lower mortality than a sardine, and so the initial conditions are going to contribute for longer to prediction scale than for a sardine. And then again, this is contribution of fishing mortality, but actually knowing what the catch is going to give you a lot of information about what the biomass is. And this is just a plot that sometimes is done in stock assessment to see, how good is the stock assessment? Do you understand the relationship between fishing and the productivity of the stock? And for Pacific bluefin tuna, she's been fished down quite a bit, so now we have less than 10% of the spawning stock biomass in an unfished state. This is a working paper that WHOI put together from the Southwest Fishery Science Center, but just showing that if you are over here, let's say in 2008, and you generate just a forecast of what the biomass will be, 10 years from now, or from year 1 to 10, and you're keeping all the parameters to sort of their mean conditions, so even recruitment, just the mean stock recruitment relationship, but you do all the catch, and then again, we don't have any observation of fish biomass, but we have these CPU indices, and then from that biomass model, you derive again the CPU index, and you have the actual CPU index here, which is in these dots. You can see that there is some scale. It's obviously not perfect, but you get the tick in biomass, which was brought on by just reduced fishing pressure. And then just, I wanted to let you know a little bit about how these projections are done. And recruitment is generally sampled from the historical distribution. So you have random variability in recruitment, and generally other parameters like mortality and growth are kept constant. And so I looked at, okay, what if you actually would have an idea of what, instead of just having random recruitment, a seasonal forecast, if you have a strong relationship between recruitment and an environmental variable, could give you an inkling of what the future recruitment is going to go. So in my paper, I look at sardine and a relationship that is used in the harvest control rule between sardine recruitment and temperature, and basing catch advice on that forecast of biomass informed by a recruitment forecast informed by a seasonal SST forecast is better than the status quo. What do you mean is better? Is better in terms of the management objectives that are important to the managers, like biomass or catch. But I want to stress that the results are really time dependent, and so the forecast accuracy in terms of the SST was too low to be useful in leads of 5 months or greater. And so just concluding with sort of some future needs, and this speaks to the sardine example because now we are revisiting this SST relationship and expanding on it, but there is a need for strong mechanistic understanding of this environment-fisheries relationship, especially if you're using statistical models, and so there has to be an iterative cycle where you keep checking that these statistical relationships are holding. Focus on what is useful, and so I guess then I need to stress that co-development with end users is key, and to figure out what the appropriate spatial scales are for the processes of interest, as well as what the variables of interest might be. And then just improve accessibility and delivery of forecast output is really hard for a fisheries scientist, of course, to access the NMME and downloading all the data from there. Skills assessment, we've seen, with some of Mike's work of variables other than SST, but also of the actual ecological forecast. There has to be more of that is done. And long retrospective forecast to actually test the reliability, and long reanalysis for training. That's it.
I was wondering if you could comment on, now that you had some collective experience with a number of stocks, what's the partitioning do you feel between sort of the natural memory and the life cycle of the fish and those environmental signals? You had that kind of idealized picture from the paper with Mike. Put some rough numbers on it based on your experience. What have you been finding?
So I would like to look into that more. Maybe you can get a postdoc to do that. But so definitely it all depends on the life cycle. So if you're a ground fish, you're gonna have a lot of that skill. So maybe a seasonal forecast of temperature, actually SSH, for instance, might be 40% of the species or bottom temperature is not gonna really provide you much more than what the biology is providing. I think for something like squid that is, faster lived species, that's very different, or maybe sardine is there in between. And but then that's different, of course, if you're moving more in the interannual timescales. So then that's why I guess multi-annual forecasts are important in this case, because there is a lot in terms of the seasonal forecast skill that you can get from the biology itself.
I could ask one more. So I was also curious, what is your sense of your optimism or not of how much traction these sort of environmentally informed anticipatory approaches have been getting in terms of making it to the decision-making process?
Yeah, so I don't think we were discussing in our little discussion group, so how many of these have actually influenced real decisions, right? And I couldn't really think of many examples. I mean, the Nowcast, maybe with the sardine survey, right, one, and EcoCast, maybe if the fishers are actually looking at them. But in terms of the sardine, I think now with the downscaled seasonal forecast, we might have a longer lead time, but just the 4 months was not enough really in terms of the process, like the stock assessment process. You need a lead time of at least 6 months or longer for that to even start getting incorporated into that process, which we didn't have at that time, but we are also revisiting the SST, sardine relationship. So yeah, I think there needs to be a lot more work just spent talking to the council and to the people, but so you need dedicated people to be able to do that. And also, I think just even doing more work with the stock assessment scientists themselves. And I think they're open, but there is a high bar to cross. Like you have to show that it's a better model than what they're currently using than just the random forecast.
Sorry, I think we should probably move on, but hold the thought, please. That was actually really great. So please, let's have some more discussion on this. I think it's super exciting to see these longer timescale popping out in the fisheries. And so now we have Sam Sidlecki who's going to talk about hypoxic conditions, or predicting hypoxia. I hope. It's your slide.
I was like, that was Emily's slide. Okay. Okay. Can you guys hear me okay? Wonderful. So I'm Samantha Sidlecki. I'm an assistant professor at the University of Connecticut, and I'm going to talk to you about work that I started on during my postdoc at UW at what is now called Seacoast, but was formerly known as JSAO. And primarily what led into this prediction system called JSCOPE, which is JSAO's seasonal coastal ocean prediction of the ecosystem. There's been conversation about changing the J over because it's now Seacoast. But in any case, we talk about seasonal forecast of ocean health is primarily what I'm going to be talking about today. And this is, of course, work like has been pointed out previously with a large team. Let's see if that one will work. And the kind of group that has been on multiple different, so like the core JSCOPE team that's been involved on multiple different applications is really talked about here. That's Isaac and Al, and Nick, and Al's online, I think. Simone maybe as well. Emily is going to be talking has a poster here, and I'll show some of her work in a sec. But there are many others, and I'll get to those other collaborators on the specific applications as we go through them. So we can't talk about today without showing Desiree's slide. So in keeping with that theme, I decided to put this as my opener. There's some specific things that are a little bit different than what other folks have said. Of course, I agree with everything else that's been pointed out about this slide. But in particular, that the forecasts are on whatever time scale you're looking for, have to be tailored to the particular stakeholder's needs. And that each of those stakeholders and managers requires specific variables or indices or outputs to make and support their decision. And in our experience, it's important to test the model performance specifically on targeting those variables in order to build trust with those stakeholders. I'll talk a little bit about that. And of course, there can be societal benefits if the model has skill in those performances. So the forecast system, JSCOPE, you can find out more about us on we're posted on the regional IUS portal NANUS, and so there's the web link up there. So how we built the model, it's downscaled from the global coupled simulation called the climate forecast system, and that is very coarse resolution, and so we downscale that with the UW Cascadia configuration, which has higher resolution and has now all the way up through carbon chemistry. And then that dynamical model is then, taken to these habitat models, or ingested into habitat models, like the ones that Desiree was just pointing out. And so there's been applications for sardine, for hake, juvenile crab, and an adult crab paper that's in the works that Emily's poster is about, and I'm sure she'll talk more about during her highlight later. So here's kind of what the website looks like if you were to go there. I pointed out for a couple of things. One is that the forecasts have been up there, true forecasts, since our first one was January, initialized in 2013. So we're coming up on our 10-year anniversary of putting these ocean forecasts out on the web. Each of those years you can go back and look at, and there's like below the times, you can see like the different pages that we produce with output, specific for with specific stakeholders in mind for each one of those forecast years. And then there's all sorts of other information there on the side, which I'll refer to more in a sec. And the other thing is that we are, which says here, is that we are all of these products then are delivered to the Pacific Fishery Management Council each spring as part of the ecosystem status report. Okay. So the performance testing with stakeholders in mind, using the data they trust, is like something that we have focused on over the years. And so this is a map just showing you like all the observations that we have used in order to do that. And what I mean by performance is kind of like the ability to produce the like seasonal trends in the region. And we compare the observed and simulated climatologies or historical conditions, as it were. And so when we work with the OA cruise planning, we definitely use their the comparisons to the West Coast ocean acidification, historical cruise line. When we work with the groundfish fishery, we definitely work with like all the small dots there in the background is like the groundfish trawl, comparisons to their benthic data. When we work with the Pacific Fisheries Management Council, in that ecosystem status report is always the 25-meter aragonite saturation state from Newport line, right? And so, these are observations that those groups have been looking at, and that we make sure to make comparisons to in a direct way. So, for example, for carbon, this is an example of over many years now with the historical comparisons, and they're all grouped together, but that we do a pretty good job historically with DIC, although there's room for improvement. With the coastal tribes, we've also worked to connect, and so this figure, it should say, it does have from PMEL and Simona Len working with the Center for Environmental Visualization. You can get that bird's eye view of the Olympic Coast National Marine Sanctuary, and those triangles there are moorings that are just off the coast of the tribal communities there located nearby. And though they look at that information, they look at the moorings, and so it is essential for us to make comparisons in working with them. So we did that in 2016 with both the climatology as well as from the observations from those moorings. This one happens to be off Cape Elizabeth in 42 meters of water for surface and bottom fields, including the biogeochemistry. In terms of uncertainty, I swear I had this slide in here before the conversation this morning, but we have a mini ensemble, which is just only one measure, all right, of all the aspects of uncertainty that were talked about earlier. And so again, one way of conveying that information, because the stakeholders are used to looking at Cape Elizabeth, regularly, and they know and have this intuition about when hypoxia develops, and from looking at the observations year after year, and they're out there on those waters, right? Right there, we provide on the website like what our little ensemble looks like for those particular locations. So it's a forecast at that mooring location, and the timing of when hypoxia occurs is for each one of the ensemble members, as well as the climatological average, are shown in the dotted lines there, so that we can convey whether or not it's likely to be earlier or later in that location for hypoxia to develop. in terms of our own going back and kind of evaluating, we find that the January forecasts tend to predict the hypoxia in this location about 10 days earlier than observed, and that the April does it about a day later than observed over the many years that we've done this so far. And so that's another way that we can kind of like bring in our like confidence in the model to uncertainty conveyance with the stakeholders. The PFMC materials that we deliver every March include the Hague forecast that was talked about by Desiree, that then it also includes, this is particularly the January 2021 forecast for aragonite saturation state along at the bottom. And it's not delivered as an anomaly because that's not what they want. They wanted the actual fields to look at where the threshold of 1 is, on the bottom. And then we write up a little blurb like what you see on the side there with some explanation about it. And then you can also find the product on our website. We also provide similarly something like that for hypoxia. So we do continued evaluation and trust building through a year in review exercise. So it's all well and good to put the forecasts out there, but then how did they do? And so on the tab on the left, you can click on year in review and look and see at that same mooring location. This is because I'm following through the example, but there's other locations that are on there, kind of what happened observationally and what the model did. Now you can see that the model has a tendency to get anoxic, especially in the forecast by the end of the summer. And that's like a known weakness that we write about in there as well. Coming back to the theme this morning about some of the atmospheric products that would be great to have. The end of the upwelling season is very difficult to forecast on these timescales, and so that fall transition, which happens very abruptly in the Pacific Northwest, as anyone who lives there can tell you, that summer ends like that unless there's a storm system that moves in, and that flushes the oxygen out very readily, and if we don't capture that well in our forecast. So we also have a disclaimer about that known bias in the model, in the forecast themselves, like on the website. So predictability is a different skill measure that was brought up this morning in Mike's Jaycock's talk, and stakeholders want to not just understand that we can show that the product works, forecasts work, but that we know why. That also helps to build their confidence in the product. So we do explore this as well. This is one from that 2016 paper showing where we showed the anomaly correlation coefficient, plotted spatially. This is a sea surface temperature, and bottom oxygen. And what the main finding here was that the subsurface fields do better, right, than the surface fields. But now we've gone back to 1998 and separated them by ENSO conditions. And I added now bottom aragonite saturation state because we have that now, which we didn't in 2016. What's interesting is that subsurface still remains skillful. You can see bottom temperature, but that bottom aragonite doesn't really seem to be doing well during strong ENSO events. But if we look at the neutral conditions, they look much better. So bottom temperature remains strong in both states, but aragonite saturation state looks better is more skillful in terms of this predictability measure in neutral conditions. So this is new and something that I'm still chewing on, and I'd love to talk to you all more about this. I have some ideas in the next couple slides. Just to clarify though, this is from the long run from 1998 through 2017. The N has come up before in some of our discussions. There's only 6 of these for the neutral conditions. And this is ACC of the January forecast in the summer upwelling months. So this is at the tail end of our predictive, this is like a 5 to 8 month time period. So it is a very difficult, and the points about the spatial averaging and temporal averaging that were made in prior talks are also real, right? So this is a very hard test, what I'm showing you now anyway. There's ideas about why that might be. So Slagna is here, and this looks like it got cut off. Sorry about that, the words there, but I'll just tell you that, and she'll tell you more about it later on her poster. She's talking about a paper that she has in review now, where she analyzed CFS, and one of the things that she found was that the summer subsurface temperatures were highly correlated to the subsurface temperatures from the winter prior. And so what you're looking at here is that correlation between those two time periods along 26.4 from the CFSR reanalysis. And the first panel shows you all years and then broken up into ENSO and ENSO neutral. And this particular analysis goes back to the 70s, so further back than we did with our downscaled experiments. And you can see that the ENSO neutral years have much higher correlation there for bottom temperatures between that subsurface and the winter prior. That's stronger. Even though the correlation exists in both, ENSO and neutral, it's stronger in the neutral states, which is interesting. And points to that reemergence idea that Mike Alexander talked about this morning. And she has some ideas about the role of that versus advection between those different states that she'll explore in her poster, so you should check that out. So we worked on taking this to the crab fishery because their subsurface conditions, that benthic habitat, was something that we found had more skill and the variables of interest to the crab fishery. So we had a project funded by NOAA MAP, the NOAA MAP program, our advisory council included the regional managers, including the tribal nation. And of course, Dungeness is important because it's an economic powerhouse, but it's also culturally important to the tribes as well. And then just, it also has a complex life cycle. So part of it is benthic, but then part of its life stage is also pelagic, something to consider. So the crab fishery in Washington and Oregon is a state-run fishery. It's not a federally managed fishery, co-managed with the tribes. And so this is figures from Emily's upcoming paper. I should include that citation there, just detailing different decisions that the managers are making and the role for the seasonal forecasts in them. And so, in the fall, they start doing a test fishery, and then using that test fishery, they decide when the crab fishery will open and whether or not there'll be crab for Christmas. And the opening of the tribal fisheries in Washington happens first, and then Washington, Oregon, the state fisheries open after that. And so they're deciding about where to open and how much to allocate together. And then once the fisheries open in the summer, then is when the hypoxia happens. And so that's when the late summer is at the tail end of their fishery season when they experience closures potentially due to hypoxic conditions. But then, of course, the fishers are also themselves making decisions about when and where to fish all along that time. Okay, so can these ocean conditions from JSCOPE forecasts predict the spatial and temporal variability of the Dungeness crab catch? This is something that Emily Norton has been working on, and she has a poster here that you can hear more about this by talking to her. But she developed, as leading with Isaac, a model to model the Dungeness crab catch in the area. And it's based on 3 different kinds of variables. One is a static, which is mostly where the fishing behavior is, the static environmental conditions. There's a dynamic oceanography terms, and then there's also the lagged conditions from prior times, and that's really supposed to mirror that complex life cycle. So it takes 4 years for the larval stages to settle and develop into adults. And so those lagged conditions are meant to represent that earlier life stage exposure. Okay. And so the model does a really good job historically. And so here's an example of that, showing you observed CPUE in the solid lines and the predicted in the dashed. So this is a combination of the static, dynamic, and lagged variables. It certainly captures interannual variability and that downward trend that you see every year. That's very sharp, because a lot of the catch is happening right after the fisheries opened, right? And she found that the ocean conditions are important drivers of that interannual variability in crab catch through a detailed analysis of figuring out, which variables are more important, which category of variables are important. You can talk to her more about that. And so because it does a good job, like I said, in the past, including when we reforecast crab catch spatially as well, When we went forward to try and forecast it, because the observations we were using were logbook data from the fishery itself, in part, the forecast of fishing behavior appeared to be necessary in order to provide true forecasts of crab catch. So she can talk to you about that as well. She's got plots of the different kinds of fishery behavior we examined, and thinking about that and trying to forecast that on seasonal timescales was not something we were set up to do prior to exploring this work. One product that we were able to co-design with the tribal and state fishers was this Hypoxia Forecast, and so in particular, they wanted to see not the anomalies, but the actual threshold for hypoxia mapped, like this. And they decided on the color scale and asked us to include locations of assets and like names on the map, which, social scientists have told me more recently, but wasn't something like place names and things like that I wasn't really thinking about in generating these, but working with stakeholders, you learn that. And so after 5 years of working with them, on this stuff, the management decision point where the Quinault Indian Nation did take management action based on a combination of observations and forecast models, both the live ocean, which is a short term, and the JScope forecast for the region, and they closed their 2018 fishery early because of hypoxia. And so that was the first time that we had seen a management action in response to our forecasts. So key takeaways from this are the forecast products that we work on from JScope were co-developed with and used by the Washington Department of Fish and Wildlife, as well as the Quinault Indian Nation, as well as the NOAA Northwest Fisheries Science Center and integrated ecosystem assessment partners. But all those partners were there from the proposal stage at the ground level, like working with us. And some people call it high touch, right? We meet with them many times a year and walk them through the delivery of the forecast, as well as other events that are going on. We end up talking about with them as well. The predictability and performance testing suggest that our skill continues to be best in bottom conditions, and our preliminary results indicate mainly for biogeochemistry in terms potentially only during the ENSO neutral conditions, and that's something to think about. And so the bottom conditions, including both temperature and saturation state, are ones that fed into the crab model. So saturation state was something that ended up being significant and included. And the JScope forecast of ocean conditions are already included in the ecosystem status report presented to the PFMC every year. And those ocean conditions that are dynamic appear to be important drivers of the interannual variability of crab catch, which was something that we learned through this exercise, which had been hypothesized before. And the model does skillfully reforecast those crab catch patterns in space and time. However, improved forecasts of fishing behavior in particular are necessary to provide a true forecast of crab catch moving forward. And so, the main point is really that there's iterative collaborations, which Desiree also pointed to, and conversations with our regional managers can be used to develop these proactive strategies informed by robust science, and that this is possible, in order to implement to age management and sustaining marine resources moving forward. With that, I'll just thank funding sources and put my contact information, and happy to take questions if there's time.
So you produce a lot, forecasted a lot of biochemical quantity, like oxygen, pH, and so on. So which type of observations you use for initializing the model, for validating the model?
Yeah, so we use whatever people will give us, honestly. I'm a massive ingester of observations. When people have them, we try and bring them along, as much as we can. And so this map shows a lot of them but not all of those have biogeochemical data. I will say all of the moorings that I talked about within of which there are many in the sanctuary, are equipped with temperature, salinity, and oxygen at the bottom. So there is BGC information there. Carbon is definitely the most limiting, especially going back in time. And so for going back further in time, we rely on these empirical models that were used on were generated on basic hydrography, that are out there in order to, evaluate things going back in time. We also developed our own version of those a long time ago to use for our boundary conditions and initial states. And so those empirical relationships are important in both ways, really. And we have different empirical models that we use to initialize the other biogeochemical fields. So each, the oxygen in particular. Does that answer your questions, Mandy? Yeah, the real-time ones are really important, though, for the trust building, because I tell people, now, well, you forecast it's going to rain, you go outside, you experience whether or not it's going to rain. Without the real time of the oxygen, there's no way for people looking at these forecasts to know whether or not it really happened, unless they're seeing the impacts of it, on the beach. Mm-hmm. Yeah.
Hi, Sam. That was great. I have a question. It's so interesting that your predictive skill for ENSO years is low, and this morning we were hearing for some variables in Mike Jacobs' analysis that the skill is higher in ENSO years. And so I guess my question is, do you think the bottom, is there some relationship between the bottom fields that your oxygen is sensitive to and the fields that he was showing that were not very well predicted that might help to explain that?
Yeah, it's a really good question. This is the newest piece, and I haven't had a lot of time to dig into yet. But I will say that the bottom temperature still remains strong. And I think if we made an apples-to-apples comparison where I averaged over the entire domain and looked at something a month lead time, 2 month, 3 month, like the boxes that Mike showed us, that it might look more similar. The way I'm showing this here is a harder test a little bit, than that. It's spatial, all that spatial information and point to point between the two. But that doesn't answer the biogeochemistry. So I think the temperature is the same, meaning the bottom temperature remains strong during the ENSO years, even though it's weaker than it is during the neutral years. I don't think Mike looked at neutral years, and he's probably on the phone and can answer that question himself. But there could be, some of the other conditions that he pointed to definitely could be culprits here in terms of contributing to it. Into why, because there's also a lot of work that's gone into historical characterization of La Nina and El Nino, and how when we know those states are different for pH and saturation state on the West Coast. I didn't show that here, but there has been work done showing that we know that there is a clear historical difference between those states, and somehow the forecast isn't capturing that. And it's interesting to me when we find this stuff, but it also means we have more work to do. Yeah.
Very exciting. Time for one.
Yeah, I just have a question about strong ENSO years. Which years?
Yeah, so we use the ONI index, and as a cutoff of 5.5.
So their definition, 0.5 is a little bit above the median. It's way more than a third.
So you think we should change it to be used?
Well, I mean, it's their official ENSO definition. I don't think that's a bad number to use,
Yeah.
but I wouldn't call it a strong ENSO either.
Okay.
I would just say it's the official definition of ENSO, which they need to do partly for non-scientific reasons. They pick that threshold.
Yeah.
And then kind of maybe related to that, and I'll just make a comment that Mike has heard me in the last month, which is predictability is not predictability.
Agreed.
It's not a measure of the skill, it's a measure of more of a theoretical, expected limit to skill, how the skill might vary in different circumstances. So I agree that, going to stakeholders and saying this is how skillful we've been is useful, but technically it's not quite the same.
They don't actually care as much about this as the science community does, right? They care about that we understand the why, but they don't ask me to see this.
Right. predictability is more about understanding the why, but I'm not sure you're showing
The why.
here. You're just showing more.
Yes.
We know in over a past period of time, which may be skewed by the fact that the 2015 event was an atypical, and so where the tropical response was not even canonical, let alone what happened next in tropical.
Yeah, we haven't gotten, you're right, I haven't answered the why. It is something that I'm curious about, and in particular, how much that, what we know about the strong ENSO, I mean, it's talked about all the time in this room, it's for seasonal predictability, right? That's connected in the West Coast, and dynamically it's very much so true. But it's possible that the BGC may have other mechanisms that are not on that list that we're just starting to scratch the surface to explore. And that's really what this, I think, analysis shows, right?
Yeah, I would agree.
Yeah.
Yeah, so I'm sorry, I don't want to truncate this, is exciting. I'll just note that Mike Jaycox mentioned that part of the difference in the period that Sam's looking at, that the predictability associated with ENSO is strongest in late winter, early spring, and in the summer, there's not so much. So
Yeah, it's part of the reason why our January forecasts do better, right, than the April.
thanks, that was good talk. That's fantastic. So next, we are going to get to hear a little bit from Clarissa. All right. I can't handle two things at once. Who's going to talk about HAB prediction? I wanted to check because I knew you could talk about so many things.
All right, I will trip. Okay, everybody. It's really fun being here to talk about halves. Hey, these are not the right slides.
Here, can you talk about those?
No. I could always throw mine up here. This is what happens when you send new versions while you're flying on an airplane. I knew there was a highlight thing that something like this happening.
So, no, I
Sorry about this.
Or is it just the one in your Dropbox?
Yeah.
Okay. There you go. Because I just wanted to know that awesome.
Yeah.
While we're doing this, did anyone want to continue that exciting ENSO discussion? Because I didn't want to cut it off.
I have a very naive question that I'm hoping somebody has the answer to. The JScope product looked awesome to me, and it's clear that someone's using it, but in the event that someone isn't using these products, I'm wondering what they're using instead. I mean, are they just, if my knees are achy, then there's hypoxia, so I close the issue. I mean, how are people actually making decisions if not with tools like this? Speaking as a pure physicist who I know very little about how decisions are made in real life. I feel like we should probably know the answer to that, maybe?
And I think Clarissa can speak to that or some, but they do tests, go out and take water samples and or take samples of shellfish for ants, for instance, right? So it's more about making a decision based on the observations.
So they have to see it to believe it, basically.
Yeah. But also they don't, but I'm not sure that they have a forecast everywhere. In the Northern California current, there is JScope, and then Sam and group have done a great job of interacting with the stakeholders to make part of this.
So would you say that a lot of decision making is reactive as opposed to proactive. Fair enough. Thank you.
Said when I was, you did a part of one of your predictive skills relying on fisheries forecasts, and behavior of fishermen. It struck me as kind of a nice opportunity, because ideally one would be able to give some advice as to which activities to avoid or to pursue. And so is that how you're doing that too? Like the fact that the fishing behavior maps on the outcome in a meaningful way is kind of nice opportunity.
Yeah, that's really interesting. It is. I think that we also are worried, like we're also asking that maybe we get, and I think this is not unique, this ask. Desiree would be able to say more maybe, but like for others in the room, but like some fishery independent observations of biomass seem like co-located with the ocean conditions seems also really critical if we're going to continue to try these avenues like this. And I think other fish stocks, do have that, but in this case, for this state-run fishery, that they don't, and maybe this is also a reason to prioritize that kind of as an observation in the gap moving forward. So like fully diagnose that because it's because we don't have an independent check on it. So that's the hard part about. I think that I would be interested to talk to you more about it. Yeah.
Sam, I think I know the answer to this question, but I want to make sure. Why does the Quinault close their crab fishery during hypoxia years?
I didn't say that. That's a good question. So because they put crab pots out for like 30 days, they can leave them out for a long time, I guess. That's what I mean when I say long time. And when the crab pots are in the water and hypoxic conditions develop, the crabs can't escape those conditions. And we've seen videos from the Oregon Department of Fish and Wildlife of the crab perishing because they're trapped in that. Whereas normally they'd be able to move. And the traps don't discern between juveniles or females or males, even though the fishery would never catch if they're too small or if they're female. So the fishery wants to sustain by making those kinds of decisions, right? But when they put them out when there is hypoxic conditions. So there are some groups working on like sensors for the traps, and of course these forecasts are another option there. That either the fisheries can take an observation and look at a forecast beforehand, or the managers themselves can take this kind of action and close it during the time period. We know a lot about where hypoxia develops and when.
Have the fishers considered or proposed just shortening the string soak times rather than closing the fishery outright?
I want to try and get one too, just in case.
Sure, let's do that.
Too late.
Yeah, I think we got it. Sorry. All right, let's take two. Okay. All right, Again, hello. Glad to be here talking about HABs with the correct title this time. So I'm going to kind of follow in Sam's footsteps. Thanks for setting me up nicely, Sam, with all the stakeholder discussions. We're going to get into what folks need when it comes to harmful algal bloom prediction on the West Coast, how we have ascertained that through workshops and other means. And then part of what I'm showing here on this first slide is that we've got a really robust modeling community on the West Coast, all of whom are collaborators in one way or another in what I'm going to talk about today. On the right, a lot of our end users, partners, however you want to refer. Usually these are folks we do very closely partner with in this space when it comes to harmful algal bloom prediction. A lot of what I will talk about today is Sugnichia and domoic acid, not all HABs all the time. We wish we could get there, but we're focused on certain things right now, particularly because of the Dungeness crab crisis that occurred in 2015. Another impetus for this talk is the Coastal Ocean Modeling Testbed project that Chris Edwards leads that has been a driver for thinking through some of these problems. So this is that COMT project. COMT funds this, and it surrounds the issue that the West Coast Ocean or Operational Forecast System has now become operational. We've had to think about what the impacts of that will be on a number of different ecological forecasts that rely in some way or another on some kind of a ROMS instance, real-time ROMS or otherwise, and what would be the ramifications for the biogeochemistry, the HAB prediction, the dynamic habitat modeling, a lot of the things that you've heard about today. So as I talk about WCOFs, you'll know what I mean, and it is a West Coast-wide model. We've subset it for California as I go ahead and talk about the HABs, but I want to point out that part of this COMT project is thinking about this stakeholder needs through the lens of the regional associations of IUs, which is why I'm involved in the project for the most part. Henry, Jan, so that's Scoos, Senkus, and Nanus, and we have come together in collaboration in this project to think about the modeling. We already think a lot about the feedback loop between observations, the management of those observations, forecasts, and user products, and you heard a lot about that from Sam and how that works at Nanus. So one of the things we did early on in this project was have a workshop at Mbari, and we brought 38 plus people into the room who represented different areas, different sectors of different industries that might have a stake in something like a WCOFs model if it were to become operational. So at the time we started this, it hadn't quite transitioned. So a couple of, we went through these major components that were our project, which the observational impacts of the data assimilation, the fisheries habitat component really centered around the EcoCast model, ocean acidification and hypoxia, another one that centers around outputs from the UC Santa Cruz Nemero coupling with ROMS, and then harmful algal blooms. We talked a lot about the harmful algal bloom stuff in the context of Sugnichia and domoic acid and the operational forecasts that are out there. So these are the outputs from that. We had we had a report, we had a spreadsheet with all of these things in it for our managers and folks at the NOAA end to be able to look back and see what is really needed, capture these user requirements. So I don't have time today to go into these. You've heard about the first three quite a bit. I will focus on harmful algal blooms today, and I'm just going to, for the rest of this talk, I'm going to step through these 5 points that came out of those discussions. And it's going to cross time and space scales depending on that need. And it's not just going to focus on modeling. I'm going to talk about the full arsenal of things that we have in our toolbox to sort of throw at HABs, HAB management, HAB prediction. And so the first one I'll go into is the improved lead time thing. That really came down to what we found. We didn't have a lot of requests for seasonal outlooks on the in the HAB discussion. We had a lot of requests for nowcast out to 2-week forecasts in terms of what is the sort of horizon over which a lot of people could respond and do something to mitigate. Tracking interstate trajectories, that came up. How do we look at what happens in California and whether that makes it to Washington, vice versa. Nearshore impacts of HABs, this is a big one. This is one where I think we have a lot of work to do, is getting our models into the estuaries and zones where most of this aquaculture and nearshore shellfish growing is happening. Complexity in food web interactions and surface to benthic coupling. This one is still a huge challenge. I'll talk about how we're trying to get there, where people want to know when will those crabs get toxic, what animals will get toxic. As you probably are all aware, any given event can lead to toxins or not toxins, a HAB, it's the actual impacts are shockingly disparate from one event to another. And then predicting offshore bloom initiation, I'll end with that. Yes, so the arrows do sometimes work, and sometimes not. All right, so for the first one, improving lead times. One thing we've been doing for a long time in California is just good old-fashioned harmful algal bloom monitoring. We have a program we still sustain through SCOOS and SunCOOS. This is weekly monitoring, having a human do the microscopy on a weekly basis, doing the toxin analysis, and also looking at full suite of variables that you can see there. We look at a suite of toxins, not just domoic acid. When we do this now, we're looking at nutrients. We're trying to quantify a lot of the ancillary variables that are going to drive HABs, and we're looking at a full suite of taxa as well. So in this particular instance, we are putting out weekly alerts of all of these groups that have the potential to cause harm, the potential to wreak havoc. They don't always wreak havoc, which is part of the complexity of HABs. And I'm just pointing out with each of these slides as I go through, I'm going to point out the limitations. So in this case, some of the things we want to think about is that it is only a weekly snapshot of a very highly dynamic system. Very expensive to do this. If you wanted to do it on a daily basis, we wouldn't have the ability to do that with the people power it requires. And toxins and nutrients are not close to real time. We get that domoic acid turnaround something on like, at best a week, at worst a month to 2 month basis. So that's not very helpful for end users. And people want to be forewarned, ultimately. So another thing we're doing, and this is a new project that's being spun up, and it's a collaboration with a lot of folks in California. And this comes from California funding from the Ocean Protection Council, where we convinced the state somehow to buy a whole lot of these very expensive imaging flow cytobots. There already were some from research projects in the region, but we're now at almost 11 or 12, based on the leveraging that we're doing and the state input. So it will be the biggest network anywhere. Gulf of Maine, of course, has a very nice network with Heidi being here, and we're collaborating with Heidi Sosik and Mike Brosnahan on another project that pulls all of these data into a very robust national data assembly center. That's a separate project that I'm not going into today. But this has a lot of power. It has power for modeling. The amount of data, every 30 minutes, we get the full resolution of the phytoplankton community through the images, apply the machine learning classifiers to those images, do all the facial recognition, and start to populate a database with all of these plankton. You're seeing an image there of a recent Lingulodinium polyhedra bloom that we had. That's the beautiful bioluminescent dinoflagellate that you see a lot in Southern California. But here, while this is a very robust and expensive network that I think is fairly cutting edge, there's no toxic information unless you pair this with an environmental sampling processor, which are even more expensive. Users still need a reliable forecast in addition to, I think kind of as Sam was saying, you get this real-time data, you can point to it in a validation sort of sense, but there's a lot that's missing, and you're not getting the toxins out of this, which is a big problem. So starting back in 2004, this is well before we even kind of envisioned the Pacific Warm Anomaly blob crisis, where HABs hit a fever pitch in California. Everyone was clamoring for a model. We were thinking about this as when I was a young grad student thinking about HAB prediction, and started to put the pieces together that became what is the operational HAB model in California now. And so this was kind of just this Frankenstein approach of cobbling together what we could. We did a lot of statistical modeling and habitat suitability models, many of which, of course, required nutrients to get it right. But you could almost get it right if you took those out and just used what you can get from ocean color, what you can get from a dynamic hydrodynamic circulation model. We use DynaOF to fill in, interpolate the spaces in the satellite imagery so we have a nice seamless product, merge those together, and then at the kind of pixel level, then apply the habitat models to that. And then we did this specifically to get at domoic acid. Sudnichi was part of it, but it's not a rules-based approach. You don't have to have Sudnichi in the water to have the toxins. We were really just focused on modeling the toxin because there are very few models that actually get to the heart of the matter when it comes sometimes to have. So usually it's just chlorophyll, sometimes it's a species of interest. But this was a many-year, 15-year process of sort of stepping through this and then working with NASA to cross the valley of death and identify an end user, which ended up being Coastwatch, to take this on and run it operationally. So it's now considered an operational model, whatever that means in NOS. And I'll go into all the different components that go into this in a minute, because we do make a bulletin that comes out of this. But there is a daily nowcast and 3-day forecast. And the 3-day forecast is really just based on a simple advection scheme, sort of pushing those pixels forward using the predicted surface current vectors. There's a lot of players, as you can see. There are a lot of layers. There are a lot of things required to demonstrate operationalization, et cetera. So this is what that looks like. I just pulled an image now. This goes out on the Coastwatch ERDAP, the NOAA ERDAP, but we also use it. We ingest it, SCOOS and SENCOOS, into our portals. We now have a joint portal, which I'm sort of sharing with you here a little prematurely, where you can layer anything that's in our catalog, you could layer in with those projections. And what you're seeing here is just a nowcast of the probability of a domoic acid event. So it's over a particular threshold. And then in this plot, I just pulled down the crab pot locations to show you how you might be able to overlay any number of parameters. But we have shore station data. We have lots of different kinds of offshore data, radar, you name it. So this is great because you get that forecast. It's only for one group, though. It's only for one toxin, which is a deadly neurotoxin, and it is a big problem on the West Coast. And so accuracy, I'm not going to go into all of the skill assessment we did, which often did require just pixel-level matchups with what we have at peers. Very limited observational data with which to do a robust skill assessment or capture uncertainty. It's missing biogeochemistry. Again, like I said, this was the best thing we could do at the time to get an operational model. So it's ocean color. So it's several different bands where I used to take the water leaving radiance and just do that processing myself. Now Coastwatch does it. And there's no food web prediction. So you get this in-water prediction. And then what does an end user do with it? Most of them really don't care so much about how much domoic acid is in the water or in a cell. They really want to know whether it's going to impact the animal that they care about, whether that's humans, marine mammals, etc. So with the Conti project, we're looking at the impacts of WCOFs, as I said. One of the things we do is look at the impacts with sea harm, some sensitivity analyses kind of thing. Right now, it relies on one version that we call the California 3-kilometer ROMS that we're now calling Classic. It's getting deprecated as WCOFs moves in, looking at what does that mean. But in terms of the users really wanting to track interstate trajectories, there is potential power here with this full West Coast domain to start to make sea harm along the West Coast, not just subsetting to California. Problems there, though, it doesn't really solve your spatial resolution problems because this is a 4-kilometer model. One we've been using is 3 kilometers. We have no parameterization for sea harm in the Pacific Northwest. It would be very expensive to run the DyneOF part of this for the entire domain. And there would be a lot of temporal gaps because we're limited in terms of how we can do satellite work in the Pacific Northwest. This thing is finicky. There we go. So I was referring to the bulletin earlier, and I think this is where we've tried to think about uncertainty translation for the user community. When we put the sea harm model out there, great, this looks nice, pretty pictures, spatially explicit model, all that kind of thing, but it was definitely a struggle to gain the trust that Sam was talking about. We don't have to convince the state to make real decisions off of it, but we do want them to see it as a tool that helps them guide sampling when they have to go out and sample, because their legal mandate is to sample the commercial shellfish. Everything else really comes down to an advisory, even Dungeness crab or any other fish. It's more of an advisory, and there's no real laws in place to monitor those for algal toxins. So we work with the state all the marine mammal groups in the entire state of California. We work with on a monthly basis to get their stranding data for any animal that's stranded from domoic acid toxicosis or was likely to have. We plot those for them. We create climatologies of the marine mammal strandings, plot all the HABs data that we collect, all the HABs data that the California Department of Public Health collects, and then really this comes down to me, because we're a small group at SCOOS, is just writing that synthesis for them and explaining how the model matched up relative to all of these resources. There was a closure here in Humboldt, but the model said this and the sampling said this. We often see the marine mammals will strand and are a great sentinel related to the model, because the model is capturing this real synoptic offshore variability in HABs and you see a huge disconnect or decoupling between what's happening at our piers. Through HABmap, we'll show one version of a story. Sea harm will show another version, and often the mammals will match with sea harm, but they won't match with what's happening at the piers. So that mismatch has been a constant battle in harmful algal bloom forecasting, because nearshore and offshore environments can be really different. So as you think about that nearshore component and how all of the major aquaculture operations in California are happening in these estuarine or very enclosed regions of the coast, we were focused for a while on how sea harm was doing at predicting that toxicity. We got a lot of feedback that we needed to go in that direction. And so I'm going to talk to you a little bit about a very small project that we did that I'd hope to do more of, where as part of the NASA funding that was funding sea harm, we got a little bit more money to work with a high resolution model of the Humboldt Bay, where there is a major oyster growing operation. Strangely enough, it doesn't seem to deal with the domoic acid problem very much in the oysters. We wanted to see if that was what was constraining that to some degree. So by linking sea harm as kind of a boundary condition to this high resolution Humboldt Bay model, we're able to then do some particle tracer tracking experiments to see what is the likelihood of getting up into these. So this is kind of the coastal boundary for sea harm on the left, and then trying to get up into the upper bay where a lot of the shellfish growing is happening up here. How can we even feasibly get a domoic acid problem there if you had a high, let's say, concentration of domoic acid on the coast? Of course, this is just a passive tracer experiment, but what we did see, these are the colors represent the different parts of the bay, and it's up in that northern part of the bay where the problem really was. So up here, where we're starting to see a lot of the shellfish growing. And we really noticed there with the results of this is that there's nothing really preventing domoic acid from being a problem. There may be residence time issues there. We're not sure why the oysters don't seem to pick it up. But then I don't have it here, but we then started just ad hoc measuring weekly with the shellfish growers, having them send samples, doing the toxin measurements, started to see some of the highest toxins we've ever seen in California in the upper reaches of the Humboldt Bay. And yet these oysters are never getting toxic. There's a lot more modeling we could be doing to interrogate this, but what we did so far lacks any mechanism. Has no operational components, no real biology, but there are some mysteries that are yet to unravel, and we're still continuing to sample up there and seeing very high, somewhat ephemeral, but very high concentrations of domoic acid. On the machine learning side and thinking about getting into the food web, I've been working with a group, and this was led by Chris Free, who is at UC Santa Barbara, thinking about taking the sea harm output, which we now have many, many years of, looking at that time series relative to the time series of domoic acid in these very important fisheries like Dungeness crab, rock crab, spiny lobster. And so kind of constraining that relationship using various machine learning models. And I was really struck by the outcome of that, doing a really good job, I would say, getting in sort of that 70-80% accuracy kind of realm of predicting when those organisms might become toxic. So, we went through the Hub-Muller diagram is showing you latitudinally those responses. The lines represent closure thresholds for a given fishery. We have yet to get this out there, but I think it's really promising. It's another one of these, what can you do in the here and now? Very practical. lacks mechanism, lacks true dynamic understanding, but yet could be really useful to stakeholders. Another modeling exercise we're going through with the food web interactions is to think mechanistically about domoic acid production, taking what we do know from the statistical and empirical models, where nutrient ratios like the silicic acid to nitrate ratio have proven to be very important for turning on domoic acid production in Sugnitia. And this is work I started years ago at UC Santa Cruz with Chris Edwards and Rafe Kudela, and we've spun it into a project where we took this 0D mechanistic idea of how domoic acid may or may not be produced, tested it against batch and chemostat cultures, and now we're working on a project with the UCLA team to embed that in a ROM-spec coupled model and start to look at the 3D runs in hindcast mode and relate that to known domoic acid events. I think this has a lot of promise for all kinds of interaction work, benthic coupling, OAH interactions, and the like. But it's not operational yet, and the food web is limited. So I just have 2 more slides. This one is exciting work. I'm not sure yet how we're going to use it, but another thing that was spun up with a colleague years ago, thinking about the use of stable isotopes, the analysis at the compound-specific level of amino acids. Domoic acid is an amino acid. You might not realize it's not, that's why it's called that, but there's a lot of similarities between domoic acid and these amino acids we use to trace food webs, understand the size of food webs, their placement, where an animal feeds and how, where it feeds geographically, latitudinally and longitudinally. And we're starting to see some super interesting relationships when we compare a really high domoic acid year with a low domoic acid year. Because I'm limited for time, I'll just talk about the plot on the right, where it looks like, I think I have a little call out here in green, these anchovies, which are represented here in this del N15 phenylalanine, what they're showing you is that they have really low del N values for phenylalanine. This is a sign that even this was the 2015 bloom, waters were warm. This really corroborates things that Andrew Thompson and others are seeing at Southwest Fisheries, where these were subarctic waters. So that codes, that's a tracer for subarctic waters. So these anchovy were coming in, very high in domoic acid, coming into the system, and they were coming from cold water regimes. So we're starting to look at the 2 things and start to put together a picture of how a massive event like that works. It doesn't mean that the system isn't productive, it is. And it also means that there's animals coming from strange places. And it's been really cool. I'd like to go into this more, but maybe we can talk about it more. I just don't know yet how to incorporate this sort of information into the models. And then the last thing we're doing is looking at offshore initiation of these blooms, using long-range AUVs, putting different kinds of sensors on them to interrogate the omics, because a team at Scripps has finally kind of broken down the biosynthetic pathway for domoic acid production, and this now allows us to do true transcriptomics for DA. So we're doing this to look at offshore initiation at 3 different sites in Northern California from south to north, and hopefully this kind of information in the long run will feed back and validate models like ROM-spec. I'm just not sure yet how that's going to get incorporated, but I think that's sort of the strategy when we think about omics and how it could work for modeling. So my last slide just gives you my impressions. I started out by saying, people might always get what they want, but sometimes they do get what they need in terms of the utility, the usefulness of a model. It's not always perfect. It doesn't always get you everything, but people want to have the best model they can get. You can read through all of the points I made, but I think the last one I want to say is that co-production is really great, and you need that to know what kind of products and synthesis and interpretation to give folks. But I think that in the end, our modeling strategies always just come down to the science, leaning into things that people might not need right now, but they're going to need later, and we need to think about the R&D and the science, and not just what does somebody need right now, because it does change, and they don't always anticipate those changes. Okay, thank you, and I'll take questions.
Super sorry. Can we hold questions until the discussion period? Because we will have a chance to do that. And since we have our online audience, we have to try and stay a little bit on schedule. So our next speaker is rushing to the front. Margie is going to talk about ecological applications in Chesapeake Bay.
usually that works then, and now it's not working. No? I mean, oh, this? You want to see an arrow? Okay, great. Okay, thank you very much. So today I'm going to talk to you a little bit about various different forecasting, ecological forecasting applications in Chesapeake Bay. So we heard a couple different fisheries and acidification, various different halves, and now we're going to look at kind of all of these things in just one region, the Chesapeake Bay. So over here on the right is a map of the bay. I like to use an arrow, see if I can make that work. The Delaware Bay up here, this is the Chesapeake Bay. Down here, this is Yellow Star, this is where I work at the Virginia Institute of Marine Science. Oops, didn't mean to do that. I just want to start off with a caveat that in this presentation, I'm really going to stress pretty much just for ease of getting this done at the last minute, modeling work that's been done in our own lab. But there is a lot of great Chesapeake Bay modeling work that's been done by others. And so I just wanted to put that apology out there right up front. So since this is all Chesapeake Bay focused, I just thought I would give you a little background on the bay. So this is the Chesapeake Bay is the largest estuary in the U.S. or the continental U.S. There's always debate whether the Cook Inlet really counts or not. It also has a very long coastline. I've heard it said that the coastline of the bay is the same as the whole, roughly equal to the whole west coast. So it has a very long coastline and very densely populated. So we've got a population in the watershed of 18 million, incorporating 6 states and Washington, D.C. And also, the bay is economically very important for the region. Ecosystem services derived from the bay have been estimated to be more than 100 billion annually, and this includes many, many different fisheries, striped bass, menhaden, burgeoning new shellfish aquaculture industry, and then also, of course, tourism and lots of other things as well. So, unfortunately, over the past years, it doesn't switch when I want it to. Oh, yeah, I see what you mean. Okay, careful, stand away. So unfortunately, in past decades, there have been numerous anthropogenic impacts on the bay. And so since the 1950s, nitrate inputs have doubled to the bay. This has caused an increase in hypoxia of more than a factor of 3. And then we've got pH and omega both decreasing, not only from atmospheric increases in atmospheric CO2, but also these changes in nutrient inputs as well. And in order to deal with all these anthropogenic impacts, the Chesapeake Bay Program Partnership has been developed many decades ago, and it's really there to lead and direct the restoration of the bay. And this includes federal agencies, most notably NOAA and EPA, as well as the 6 states and DC as well. There, click, okay. So perhaps not surprisingly, given all of the ecosystem services of the bay and as well as the anthropogenic impacts, many folks are stakeholders and managers are interested in a variety of forecasting over a variety of timescales in the bay. And so here I've outlined basically 3 different timescales of forecast: daily, weekly forecast, seasonal forecast, and mid-century projections. And these are all of interest in the bay. So for example, on short-term, daily, weekly forecasts, we have charter boat captains and anglers. They want to know where they can fish in the bay. They want to know where the hypoxia is. Turns out the fish kind of congregate at that oxygen at 3 milligram per liter area. So they really want to know where they can bring clients to catch their fish. The aquaculture industry and hatcheries really want to know whether they need to, specifically the hatcheries, whether they need to treat the bay intake water or if it's particularly low in pH and aragonite saturation state. And then beach managers, as well as folks, just the public who go to the beach, want to make sure that the beaches, the water there, they're swimmable and it's safe from HABs and Vibrio and other things. So these folks are really interested in days up to maybe a week. And then on the longer time scales, seasonal forecasts, lots of folks are interested in those forecasts as well. Particularly the public, those always get, we can have a seasonal forecast in the spring of what the summer holds. Folks always really are keyed in. They want to know what the summer is going to be like. But also fisheries managers really need this longer term time scales for catch limits, for example. And then finally, we have even longer interannual, interdecadal, mid-century projections. And here we've got coastal resource managers that are really interested in this information. They want to know whether their planned nutrient reductions, so what we call the total maximum daily loads, or TMDLs, are going to lead to attaining that water quality that we had hoped it would when these TMDLs were set decades ago, in spite of climate change, which we now know is affecting the bay tremendously. So today I'm really I don't have time definitely to talk about all of these. I'm really just going to focus on the shortest timescales and the longer projections. But a lot of great work has done on the seasonal forecast. So I want to just highlight this particular paper by Downscaled et al. That recently came out in Ecological Applications. And so this is a great place to learn about some of the empirical models, or an example of an empirical model that's being used to assess seasonal hypoxia forecast for the day. So I'm going to start off on the short time scales. And for this, we've developed what we call CBEFS, or Chesapeake Bay Environmental Forecast System. And I just want to highlight over here three folks that have been inherently involved in this. Aaron Beaver from Anchor QEA has really manages the system, and he is online here. Pierre, I believe, and Pierre St. Laurent from VIMS is really one of the core developers of the model. And I believe he's attending too virtually. And then Raleigh Hood has been involved from the very beginning in helping us set up this system as well. And then we've got two papers, one by Aaron, one by Pierre, and I have the citations down at the bottom here. So basically, the estuarine model is, just to give you a general feel for the model, the grid is here in the center. There's my mouse. Okay, this is the model grid. The resolution is about 600 meters by 600 meters. We've got 20 vertical levels. This includes full hydrodynamics, tides, and carbon, nitrogen, carbonate cycling. All of this is in there. But of course, the model is only as good as the forcing we give it. So we need forcing both from the atmosphere, from the land, and from the ocean. So in terms of atmospheric forcing, basically here we're forcing it with our weather forecast. So wind forecast, temperature forecast, precipitation, solar radiation, all of this feeds right into the model. From the land, we use terrestrial inputs from watershed models, so including the Bay Program's official watershed model, as well as USGS gauge data too. And then on the ocean boundary, we typically use long-term climatological inputs, but sometimes we also link to coastal models. We're working towards that way. So this is the model system, but of course none of this would be possible if we didn't have all the data we have in the Chesapeake Bay. So I know we're really lucky, I hate to say it, but we are really lucky to have all this data. We've got 35 years of data, 17 cruises a year, more than 100 stations, and with depth of temperature, salinity, oxygen, pH, and then biosamples for nutrients and chlorophyll. And on top of that regular sampling program, we've got buoys and data flow, continuous monitoring stations, and of course the river gauge data as well. So this, of course, is really the backbone of the model. This is what we've used to develop the model over many years. So we've got these inputs, the atmospheric coastal land inputs, and we are evaluating the model with the data I just showed. And with all this basically feeds into our real-time model forecast setup. So every night we have this setup so that it all runs automatically, and we get nowcasts and two-day forecasts produced, and they are displayed every day on our website there, www.vims.edu/CBEFS, CBEFS. So if you are on your phone, I am assuming that you are going to go take a look at the website. Please do. Everyone can out their phone and start texting. No. So they're all displayed there, and the idea here is that it's really we've worked hard to get it to be mobile friendly, so that folks on their ships and can on the fishing boats can easily look at our forecasts. So additionally, we are also working with Maricruz, our IU's regional association, and the forecasts are also there in much more depth on their Oceans Map portal, and there you can do fancy things and zoom in and out and model data comparisons and also download all of our NetCDF data if anyone wants to play around with it. So we find that these two different portals are really kind of serve different audiences, different purposes. So are both useful places that you can go. So if you take a look and go to that website, you'll see something like this on the right. And if you go on your laptop, you'll see something that looks more like this. But I just wanted to, on the right here, mention the different things that you can't read over there. We are forecasting hypoxia and also the dead zone size, essentially. Various different acidification metrics, pH, saturation state, alkalinity. We have Vibrio forecasts up there, as well as temperature and salinity, and we've got some HABs forecasts coming soon. So I just will show you a little bit of a few maps. This is what I downloaded a day or two ago. So this is what bottom oxygen looks like on the bay, what was forecast to look like on the bottom of the bay today. So all blue, that means healthy waters at this time of year. No problems out there yet. Hypoxia here is completely a summer phenomenon. If you saw yellow and green, you'd be moderately low oxygen, red, very low bottom oxygen. So more interesting time of the year, this is an example of what it would look like in the middle of July. This is from July 8th, last summer. So you can see what folks call the dead zone, this hypoxic region of water being the red stretching through, I'm gonna give up on the arrow, sorry. Down through Maryland, and up into the Potomac as well. So in addition to hypoxia, we also have forecasts there on bottom pH. So here on the left is an example of bottom pH, and on the right, surface aragonite saturation state, just kind of to show you the kinds of things we have on there. And the newest thing we've added is the percent chance of encountering Vibrio. So this was our forecast again for today, and here the blues represent very low chance. So if you're swimming in the bay today, you've got very low chance of encountering Vibrio. But of course, that's not always the case, so it's clearly not a concern this time of year, but in the summer, it really can be. And so here you can see in the Central Bay where the higher, much higher chances, 70 to 100% chance of encountering Vibrio there. And then in addition, we have a time series of the percent of the bay with a high chance of Vibrio encounter. So here the blue represents a 50% chance and the red the 70% chance. So again, you can see this is just really a summer phenomena with very 0% chance at other times of the year, for instance, right now. And so we talked about Vibrio, and I just wanted to say that, again, as I mentioned earlier, HAB forecasts are going to be coming soon, and Dante Hormans is our new postdoc working on this, and he says Spotlight talk tomorrow. Well, I wrote this yesterday. It was tomorrow, but now it's going to be today. Wrote it late last night. Okay, so so stay tuned, and you'll hear a little bit more about what we're doing in terms of HABs. Okay, so that's and I did want to just emphasize again that a lot of this has been driven, like the other speakers have said, really driven from back and forth communication with the stakeholders, and I really didn't put that into these slides too much, but really we've had workshop and focus groups. We've got two extension specialists, one on acidification and then one on more the angle, one more on hatcheries and shellfish, and one more on finfish and recreational fisheries. And we've been working very closely with them to really get feedback. And so the maps that you see are here based on actually many years of working with the stakeholders to try and see what they want. Okay, so now I'm going to transition into much longer mid-century projections. And here I'm going to highlight some results from a couple of my students. This is work by Kyle Hinson. And we have a project working with some of the managers, and basically we found that what they really want to know is how climate change impacts on the watershed, so increasing atmospheric temperature and increasing the number of storms, how that's going to affect the runoff, because they're really keyed in on reducing nutrient runoff. So they want to know how climate change is going to affect that runoff and then how that is going to affect hypoxia. And secondly, they want to know how confident we are in these estimates. So we've talked a lot about uncertainty quantification here, and that they tell us right off the bat, we do not want to hear about uncertainty. We want to hear about confidence. So anyway, we've changed the question. We want to know how confident are we in these estimates, at least when we talk to them. I think I can say uncertainty here. And so what Kyle is doing, has done here is try to answer this question. And so he's done this through a very complex system he set up here. He's looking at 5 different global climate models. Need an arrow. There we go. 5 different climate models. He's using, he's applying 2 different downscaling methods to these 5 different climate models. These produce different estimates of temperature and precipitation, which are then fed into 2 different watershed models. So then we get the various different discharge and nutrient loads, which are then fed into our estuarine model that I was just talking to you about, and ultimately to produce hypoxia forecast. So we're trying to kind of get an ensemble of results here to get at that uncertainty question. So ultimately, he's got basically this, to simplify it here, 20 climate scenarios. So again, 5 GCMs, 2 downscaling techniques, and 2 watershed models lead to 20 different scenarios, all run through our hypoxia model. So here are just, let's look just for a second at just the 5 different GCMs. So he's picked, we have actually 20. He picked the centroid, cold, dry, warm, dry, cold, wet models. So we've got these 5 models. He runs all of them, and for one particular watershed model and one particular downscaling technique, and runs these all through. And this is showing here the change in annual hypoxic volume between the 2050s and the 1990s. So ultimately, overall, well, you can see an increase of maybe 5 to -5 to plus 25% chance for these 5 climate scenarios. Now, if we look at all 20 climate scenarios using the different downscaling techniques and the 2 different watershed models, basically 80% of the climate scenarios are projecting an increase in hypoxia in the future. So that's we find that the managers really want to know a number. They want some kind of confidence estimate. So we were also interested, and they were interested in what is causing this uncertainty. And so what Kyle found was basically it's amazingly similar between these 3 sources. So of course, again, we're looking at these 3 different sources of uncertainty: the global climate model, the watershed model, and the downscaling method. So 40, 32, 28. Overall, take-home messages. These are all really important. And I think we were perhaps most surprised about the downscaling technique. Is really introduces almost as much uncertainty as the global climate model. Okay, so I know I'm going through this. Hopefully that paper will be out soon, and you can look at all the more detailed analysis there. I also have another student, Colin Hawes, that's also trying to work with managers and try to use our modeling system to answer some of the questions that they have. And so he's, one of the questions that we were asked was, how does this impact of changing runoff on hypoxia compared to other direct effects of changing atmospheric conditions? So again, what I just talked about was the impact of the air temperature change on the watershed impacting hypoxia. But how does the actual changes in air temperature impact hypoxia? And where in the bay are the greatest effects going to be seen? And so here he's done a number of different scenarios trying to tease apart all of the different aspects of climate change. So here he is looking at the change in bottom oxygen between the 2050s and the 1990s this is a climatological, so the average of 5 years here on the x-axis, and the black line represents all these different climate change impacts at the same time. Green is just the impact of air temperature change, wind change, Blue is precipitation, solar radiation, and this yellow here is basically what we just looked at, because it is the impact of changing runoff due to climate change. And so the take-home message here is really that the direct impacts of atmospheric change on the bay are actually much greater than the indirect impacts that occur from the changing runoff due to climate change. And this is just shown in another way here, where these various different bars, again, the green bar representing the atmospheric temperature change, and that's really what's changing hypoxia and oxygen concentration in the future. So where is this occurring finally? So here are a couple maps in the far left here. These are results from basically our baseline simulation from the 90s. In the center are the 2050s. And then on the right is the difference. So you can see that the biggest change is really in the central bay here, with slightly less changes in the north and the south. So anyway, this is my final slide, just to conclude here. You're asked to think about different time scales of time horizons of interest, and which are of most interest in the Chesapeake Bay. And it's really hard to say because this really depends on the stakeholders. And there is interest pretty much, you name a time scale and someone is interested in it. Very different applications, of course, the daily being public going to the beach and the anglers fishing. Seasonal, maybe more fisheries managers, and then the coastal resource managers will be more on the longer time scales. I didn't here today, I really just focused on our mechanistic modeling, but again, we're working and others are working a lot on empirical modeling and machine learning, and there's a lot of great opportunities there as well for some applications, particularly potentially the short-term habitat suitability models and short-term models. And what data and inputs are needed? What do we need most? This is, I think, is one of the breakout questions that we're going to be going off to next. I would say it's one of our perennial issue is our model is only as good as what we have driving it, and this largely comes from the land. What are the nutrients coming off the land? And that's something that we're continually. We've got a lot of data. Got the USGS gauges are great, but we need they don't give us all the information we need. And then in the atmosphere, of course, we need higher resolution weather forecasts would be great. We can't go out, wind forecasts may only be good for 10 days into the future, so we're not going to be able to predict farther because the hypoxia is so dependent on those winds. And then downscaling, and downscaling is something that clearly at these scales is incredibly important, and they need some further research as well. So I will end there. I didn't go too much.
So that was great because I think that really bridged nicely some of the questions that Clarissa raised between the open ocean to the nearshore pier challenges, right? I mean, these are all the pieces that come into play in the nearshore. I'm sorry, everybody here needs a break. I need a bathroom break. And so, can we write your questions for Margie down so that we can bring them up in the discussion after the spotlight talks, please, because don't forget them. Okay, sorry. Oh, and does everyone come back?
We'll be back here at 3:30, so we'll be back on time.
Same time.
That's we have 8 minutes.
No. How does it work? I want you to do whatever it is that's hard. Hold on. Folks, we kind of need to get started, sorry. Very short break.
Yes. Oh, yes.
Oh, okay, but we're sharing the class here.
Something's wrong.
We're sharing slides.
Why are we getting an echo?
For this homework.
Oh, what? How did? Well, okay. That's right, Emily. You're right.
Yeah. Isn't it? It's only when we're in here.
Yeah.
Yeah. Yeah, it's like critical body mass.
So once Emily starts talking, we should Emily, you can hear us, right? Huh? Yeah. Oh, I can hear us. So.
Hey, Emily, can you hear us? If you are, you can unmute, and we're ready with your first slide.
Yeah.
Yeah, seacoast. Seacoast,
So if you can unmute yourself, Emily.
Yeah. She's not out there anymore.
Oh, maybe she had to do permissions. Almost ready. Thank you, guys. Okay. We'll start your, we'll come back to you, Emily.
Sorry.
She fell off the. So.
Okay.
And we need to move this, so.
For you, yeah.
Okay. Given my height. Okay, so the Northeast Pacific has been experiences, so it's prone to have conditions of intense warming, also known as marine heat waves, which have been particularly pronounced over the last decade. And they've had also some significant ecosystem impacts, so that it's important to try to predict these conditions. And so, the figures on the left show the composite evolution of marine heat waves, so surface temperature and sea surface height, starting from 12 months prior to the onset time when the temperature passed a given threshold. And so you can see that the development of this condition are associated also with the large-scale anomaly, and particularly with the development of warm condition in the central equatorial Pacific. And so this connection with the large scale suggests that there may be large scale anomalies that can lead to can help anticipate the development of these conditions. So this study was an attempt to identify these initial conditions that optimally lead to the development of Northeast Pacific marine heat waves. And for this purpose, we used the linear inverse model, which is an empirical model that is derived from observation that includes both a deterministic component, the fast term on the right-hand side, and also stochastic term. And in particular, for the purposes of this study, it also allows to identify initial conditions that really lead in an optimal sense to desired or final states of interest. And in this case, we used as a final state what is shown on the bottom right, which is the sea surface temperature at north of 25 north at the time when the marine heat wave, at the onset time, so when we passed the threshold that was chosen in this case. Now we have to move forward. Okay, so we did find initial conditions, which are the ones shown on the top in the blue box for these two variables that we chose to characterize our states. And in the panels below the blue box, you see the evolution of these fields at subsequent times. And you can see that, in fact, these conditions are associated with the warming in the Northeast Pacific, and also are associated with the development of ENSO precursors and then the development of the Central Pacific El Niño conditions. And also, this methodology allows to diagnose the dynamical operator of this model. And so we investigated which particular modes of the system, climate modes dynamical modes are more important for the development of these marine heat waves. And we found that there was one specific mode that seemed to be particularly relevant, and this is associated with the anomalies both in the Northeast Pacific along the coast, west coast of the US, and with this also anomalies in the Central Tropical Pacific. And we ended up defining it or terming it the NPCP mode. So the conclusions for the purpose of this workshop, I have three major points that I would like to leave you with. The first one is that the Northeast Pacific marine heat waves are associated with this large-scale climate variability that links the North and the tropical Pacific. Also, the Central Pacific El Niño condition develop concurrently with this Northeast Pacific marine heat waves and can increase the persistence of the marine heat waves. I haven't shown it here in these 5 minutes, but Tong there is a talk down the road that she's going to show that. So we have discussed this morning about the importance of ENSO for lending predictability along the west coast. However, this shows how not all ENSO events are the same, and so the type of event is important, and also the specific evolution of the events also can play an important role. And finally, from a methodological perspective and in anticipation of tomorrow's session of modeling, I think the LIM, which is based on observation, can be viewed as a form of machine learning, can be actually a very powerful tool for both identifying optimal precursors. It's very useful, can be very useful for predictions, and also can allow this more diagnostic analysis that can maybe more transparently reveal the underlying dynamics. That's it.
Awesome. We have Jonathan Evanilla.
All right, what's up? I'm Jonathan. I'm from the Bigelow Lab in Maine, and today I'll be talking about an ecological forecast we've been developing and delivering to stakeholders at a weekly timescale. So in the Gulf of Maine, there are annual blooms of a harmful algae called Alexandrium, and during these blooms, subsequently, there's biotoxin called PSP, or paralytic shellfish poison, accumulation in shellfish. And it's serious, it could kill you if you eat these toxic shellfish. So Maine Department of Marine Resources monitors and manages these fisheries by sampling a bunch of sites across the Maine coast for both Alexandrium abundance, this is a shore-based water sample, as well as they test shellfish tissue for PSP levels. So the goal of this forecast is to predict those shellfish toxicity measurements at least 1 week ahead of time. And this is to aid two types of stakeholders or users. So one, the fishery managers, Maine DMR, who are directing sampling each week, as there are over 500 sampling sites, but they can really only include about 10% of those per week, as well as anticipating harvesting closures and reopening during toxicity. And then the second type of stakeholder or user are industry members. So shellfish growers, harvesters, and dealers who are making business decisions on a weekly basis during these blooms. I really want to highlight this idea of knowledge co-development or co-production. Desiree has mentioned it. It's been a theme of this session. This forecast we've been developing, the stakeholders have been involved since as early as the proposal phase, and we have a very close, tight-knit working relationship with them. So we're making site-specific predictions each week using a machine learning-based classifier. And we're using a binned toxicity classification system, which you can see in the table in the center. The highest class, class 3, is representing closure level toxicity. That above 80 toxicity units is the FDA regulatory limit for when these fisheries need to be shut down. The classification is based on a probability of each of the 4 classes. So rather than just reporting our predicted classification, we're also delivering the probability of each of the other 3 classes that aren't predicted. We're only using toxicity data as input into this model. There's no Alexandrium abundance being used to predict toxicity levels. And each prediction has a start and end date, which is based on sampling that takes place roughly weekly. However, all the sites aren't sampled on the same day, and sometimes they're more or less than 7 days apart. We've also developed an interactive web page where users can go scroll through on a map of the coast of Maine, find their site if it's included in sampling, or sites that are near them to make their decisions accordingly. So we trialed this forecast with a pilot season last summer, and the forecast did pretty well. So you can see in the confusion matrix in the middle, correct predictions follow that diagonal going up the middle. So out of 533 predictions, we achieved about 94% overall accuracy. And we also successfully predicted 2 of 2 closure level toxicity events. However, the PSP season wasn't very exciting. There were only 2 of these closure level events. On a very toxic year, you could see up to 50 or 60 of these. And one last thing I wanted to point out was that in machine learning, especially classification, the more representation you have of each class, the better that class can get predicted. But as you can see in the histogram on the right, our class 0, or the lowest toxicity class, dominates the training set. So our model is really good at predicting the 0 class, but it starts to fall off as we get up the classifications. And one last thing on the left, the scatter is showing the probability we predicted of each of the predictions from last season, the probability of class 3 or closure level toxicity versus the actual measured toxicity the next week. And you can see the regulatory limit at 80 and the 2 that we successfully predicted, but then we also had 3 false alarms. So check out our page. The page is being made available to the public this season, still in an experimental phase, and it still comes with a disclaimer that this forecast should never be used as the basis of decision making, at least yet. Thank you so much.
Hold your questions, please. Don't forget them. And we have Dante Horstmans.
All right. Hello, everyone. Indeed, my name is Dante Horstmans. I was already introduced by Margie earlier. So, do I have a pointer? So it's written out here, Dante Horstmans. So yeah, and I can assure you that my presentation is right now and not tomorrow. So that's great. But okay, so I recently started as VIMS as a postdoctoral researcher, so I'm very excited about that. And today I will really introduce you to the project because I just started. I don't have that many results. It's too preliminary. So let me start with the team. So I'm very pleased to be part of this amazing team. So that's me here. It's an old picture though, and Margie already showed Aaron, Raleigh and Pierre, but there are also other people as presented here. And let me start with giving you some information on my background. So I have a bachelor and master's degree in physics, so theoretical physics, and I did my PhD at the Department of Biology in Antwerp in Belgium in collaboration with Delft University of Technology in the Netherlands. So I really combined this theoretical background, this physical theoretical background, with the expertise of the biologist I was directly working with. But it was quite exciting. So my work was very interdisciplinary, and I combined theory and application. So it totally makes sense that I'm part of this team because also here, as we already discussed, there are a lot of fishes, biologists, and also in this team we have biologists, fishes. So it's very interdisciplinary. So it makes sense. So I'm very happy to be here. Okay, so my second slide is on the project itself, a very brief introduction. So as mentioned by Margie, we have this forecasting tool. It already exists, and it predicts specific physical, biochemical parameters. Here are some examples: salinity, temperature, oxygen, pH, and so on. But what we want to do is we want to extend this tool. We want to extend it with forecasting of harmful algal blooms. As Margie mentions, it's important for stakeholders to know where anoxic conditions will occur. But similarly, this is of course not complete. It's a very good start, but there are also other variables that may be important, like our harmful algal blooms. So that's really the objective of this project. Okay, so how we will do that? That's the last slide. So what we will do is we will start with this existing list, these forecasted values of salinity, temperature, and so on. We will use this as input for empirical habitat models. And we already discussed this, but let me explain it again. So here is an example. What is an empirical habitat model? So here, this is a two-dimensional example. On one axis, we have salinity. On the other axis, we have temperature. And this model, so given these two values, it will predict the occurrence of, in this case, sea nettles. But of course, you can extend it using to multi-dimensional space, so more than two variables, but I don't have to explain it here. And the last thing is how we will do that, how will we construct such empirical habitat models. What we will do is we will use the long-term in situ observations in the Chesapeake Bay. And so we have a data set going from 1984 up to now, so we will use this long-term data set. It's also covering almost the full spatial scale of the Chesapeake Bay. So this is very nice. And we will combine this data set with machine learning techniques and statistical models, as already discussed earlier. The techniques I started with are support vector machines and logistic regression. But what's important here is once you have constructed the empirical habitat model, we will use the output of our mechanistic model, this forecast. We plug it into the empirical habitat model, and we will have a forecast of harmful algal blooms. So that's the main idea. So with that, I would like to thank you for your attention. I'm happy to answer questions later, of course, but you can always contact me here. So here's my email address, and yes, thank you again.
Jeonju, correct?
Hello everyone. My name is Tong Tong. I'm currently a postdoc at NOAA PSL at Boulder. Today I'm going to talk about the continuum of Northeast Pacific marine heat wave and their relationship to the tropical Pacific. And I would like to acknowledge my co-authors, and they are Matt Newman, Antonietta Capatonti, and Manu Di Lorenzo. So the Northeast Pacific marine heat wave, especially the blood event that happened in the 2013 to 2015, has devastating ecological impact. And this event's evolution is shown on the left column here, and is featured by a large one anomaly in the Northeast Pacific that persists in 2013, and then it extended along the California coast in the 2014, and lastly, it ended up with a strong El Nino event in the tropical Pacific. So the motivation of this study is that the 2013 to 15 Northeast Pacific marine heat wave is not fully understood, and specifically we want to answer, was this a truly unique event, or is it just an exceptionally intense expression of similar recurrent marine heat waves? And also the other question that we would like to answer is, was its intensity and multi-year duration driven by internal extratropical process, or did the tropics play an important role? The first thing that we did is we identified the Northeast Pacific Index, which is shown by these gray box here, and we used the spatially averaged time series from the observation data set to quantify the frequency of marine heat waves as a function of their intensity and durations, and that's shown in the panel A here. And so this shows that the Northeast Pacific marine heat wave happen more frequently happen less frequently with increasing duration and increasing intensity. And the next thing that we do is we develop the linear inverse model called the LIN to try to simulate large samples of Northeast Pacific marine heat waves. And we also reproduce the same plot as panel A, and it's shown in the panel B here. And which shows that our linear inverse model reproduces the statistics of the Northeast Pacific marine heat waves. So that shows that the LIM is capable of reproducing these events statistics, and it also, because it provides a large number of samples, so it also allowed us to analyze their relationship with the tropical Pacific. And we did this by first extracting all of the events for a certain duration and certain intensity, and do a composite, and then extract the NINO 3.4 index and the Northeast Pacific marine heat wave index that were defined earlier, and plot here the concurrent evolution of those Northeast Pacific index and the NINO 3.4 index here. So for example, if you look at one of the blue curve, so it shows that when the tropical Pacific is relatively cool, these Northeast Pacific marine heat waves starts to ramp up with increasing intensity. And the next phase is, as the Northeast Pacific marine heat waves stay relatively warm, really warm, the tropical Pacific starts to increase their intensity. And last, the next phase, which is the top here, is that the Northeast Pacific marine heat wave have decreasing intensity, and also the tropical Pacific is starting to decrease as well. And we do this by multiple different durations, and that shows the colors from the light green to the blue, and this really tells us that the tropical, the longer-lasting northeast marine heat waves are associated with the more persistent tropical Pacific variability. So the implication and conclusion from this study is that we think that these Northeast Pacific marine heat waves are a recurrent phenomenon, and we use the linear inverse model to reproduce the statistics of these Northeast Pacific marine heat waves. And specifically, when we apply it to do the forecast of the block event of the average forecast scale, which is evaluated by the anomaly correlation scale, it's about 0.6, so it's reasonably good. And also, this link reproduces the statistics. And lastly, we find that the durations of these marine heat wave events are associated with the tropical Pacific variability. That is all of my presentation, and thank you for listening. Please let me know if you have any questions.
So we'll go back to Emily Norton. Emily, I hope you're there online.
Hello, can you hear me?
Oh, there
yes. Great. Thank you all for hosting a hybrid event, and thanks for your patience with the technical challenges associated with that. So I'm Emily Norton. I'm a research scientist at the University of Washington, Seacoast, and I've been working with several co-authors on developing seasonal forecasts to promote dynamic management of the Dungeness crab fishery in Washington and Oregon. Samantha Seidlecki is working on this project, and she gave a really nice introduction in the previous session. But the work was initially motivated by this long-standing observation that the Dungeness crab catch varies widely interannually. Sorry, I'm getting a little feedback now.
You sound fine on our end. It might be your phone if you want to hang that up.
That is muted, so it shouldn't be causing. Maybe it's just a delay.
How's that now?
I think I can still hear myself. I'll turn my volume down and ignore it. So the figure on the left shows the Dungeness crab catch from 2007 through 2019 for Oregon in light blue and Washington in darker blue. And you can see between years, sometimes the catch will almost double. So we found that fisheries managers as well as fishers themselves are interested in forecast products that will help them know what to expect for the upcoming season. So our question here is, can we use modeled ocean conditions to predict this variability in crab catch? Next slide, please. Thanks. So we decided to develop a statistical habitat model, specifically a generalized additive model, to forecast crab catch. So because we are interested in forecasting catch per unit effort, the first thing that we wanted to consider was fishing behavior. So when and where the pots were fished and for how long they were fished. At these locations, we were also interested in other static environmental conditions such as depth and sediment type. That was the first group of predictors that we considered, and we termed those static. The second and third groups of predictors are dynamic oceanography and lagged oceanography. So these are both coming from JScope forecasts and hindcasts or historical simulations. The dynamic conditions are environmental conditions that coincide with the time that the crabs are caught. So things like temperature, salinity, oxygen concentration, chlorophyll concentration, pH, aragonite, et cetera, et cetera. And then the lag conditions are oceanographic conditions experienced by earlier life stages, because Shanks and other authors have shown that there is a relationship between abundance of earlier life stages, specifically megalopy, and adult crab catch lagged by about 4 years. Other lags have been reported in the literature too, so we explored several different years there. In conclusion, our best model included variables from all 3 classes of predictor variables. So we had static conditions, but we also found that adding dynamic and lagged oceanography from JScope improved our model fit and skill. Next slide, please. We found that our model skillfully reforecasts catch patterns over time and space, with a caveat that fishing behavior was an important part of this skillful forecast. So Sam showed the figure on the left, but just to recap that our model, this shows average catch versus day in season for fishing seasons from 2007 to 2015, so our model training period, with observed catch in solid lines and predicted catch in dashed lines. And we see that our model captures intraseasonal declines in catch over time, but also interannual variability. The figure in the middle shows our spatial biases in our forecast. So this is presented as a percent bias with under prediction in purple and over prediction in orange. So you can see that over most of the domain, our model tends to slightly under predict catch, but there are a few areas close to shore and close to the shelf break where we have slight over prediction, but in general, our bias is pretty minimal. But both of these reforecasts were generated using known fishing behavior. And we reserved 3 additional test years, 2016, 2017, and 2018, in this table on the right, to do test cases for validation of a true forecast. Because those years are in the past, we do have access to the observed fishing behavior, so we can generate kind of a baseline reforecast using the observed fishing behavior. But in a true forecast sense, we would have to generate a fishing behavior forecast. And one reasonable guess for that would be to use the fishing behavior from 1 year prior, and that's in the middle of the table. And then another reasonable guess might be to use the average fishing behaviors from all prior years. But when we use our 2 metrics for skill, either correlation coefficient R or anomaly correlation coefficient, ACC, we see that our model skill declines. All the boxes in red are lower skill than they are if we use the observed fishing behavior. So one need coming out of this work is a need for a better fishing behavior forecast, because that is important to this catch forecast. However, we do also note that the development of the relationships between individual predictor variables and crab catch are still informative for generating useful forecasts. Thank you, and I'm happy to take questions later.
So that was great. I think we had some really nice examples. Thank you, Emily. And some of the stuff that I kind of took away from listening to all these different applications was this emphasis that we knew we would hear on working collaboratively with the users from the get-go. Although I really like that Clarissa mentioned that we had this time to, to keep your mind open about considering other products that might not seem important right now, but that could become useful and relevant in the future. I've had that experience myself. And the other thing is this kind of maybe the challenge of moving not just from the open ocean to the coastal regime, but into the nearshore, and the difficulties of not just getting into the fine scale, but also the land forcing. So we did not get to ask Margie any questions, so I encourage discussion. You already have one? Somebody has one?
We were saying also Clarissa.
And Clarissa, excuse me, and also Clarissa. So if people were retaining those questions, please speak up now.
Yeah, one for actually one for decimate, one for margin. So for margin, so you stressed the importance of the air temperature margin for the hypoxia, and you also showed that the hypoxic condition are not the same everywhere in the bay. So what is the link between the surface air temperature and the hypoxia? So again, I'm revealing my ignorance on this topic. Yeah. Is that working? Yeah.
This is a green light.
Yeah.
You have a green light.
A green light, okay.
Yeah, so in the bay, one of the interesting things we found is that the surface and the bottom are warming equally in time. So the warming signal is so much slower than the residence time of the bay. The surface and the bottom's warming just as much as the surface. That's one thing, and it's two impacts. So the air temperature, first of all, is solubility, simple chemical solubility. So all year long, warm water holds less oxygen, so it's lower in oxygen. And then it's also in the summer, and that's a very significant effect. And then also the rates of photosynthesis and respiration, but particularly respiration and remineralization are much higher when the temperatures are higher. So that brings down the oxygen even more in the summer. I think that answers it. Good question.
Do you have a second question, Anthea?
I did one for this year. Yeah, so I was intrigued by your example of the European project where I think they got some good skill by describing salinity at different levels, and I was curious what is special about salinity, if salinity just directly affects the conditions where the species live in, or if salinity is just a proxy for a type of water masses or origin that will be important for a species.
Yeah, that's a great question. I forwarded you the paper because I'm not expert at that, but I think it has more to do with that being a proxy than salinity itself affecting the species, and it was also very much associated with temperature. So temperature change versus salinity changes. So
So I have a question for Clarissa. There she is. You would emphasize kind of the 0 to 12 day forecast window for most applications. But I do know that the West Coast has a really persistent season-long HABs lately. And I was just kind of curious, what would the value of a longer-term outlook be, given that my impression is that aquaculture response to a HAB is one that takes time and effort, and that there are these longer events. Do you see a role for those longer outlooks, say 3 months, if they can be done with reasonable reliability?
Yeah, thanks. Is this on? Is it green? You want green? Yeah. So it's tricky because I'm starting to, as you were asking that, I was thinking about your experience with the systems and those sorts of those seasonal outlooks for Alexandrium and the Gulf of Maine. And I think the way I perceive it from the stakeholder perspective in California is, while we might say that we have a bad year coming up or a bad season, unless you can give them a more granular understanding of crossing particular thresholds for toxins on that shorter time scale, no one is going to close their entire shellfish growing facility for a season. They're going to move the beds around to a lesser optimal place for them. So I'm thinking about the mitigated strategies and what that would require if they had a seasonal outlook. I can't think of too many economically sound options that they would have, knowing that there's a bad season, but if they know if they have a bad week, it's a lot more tractable. On the other hand, if we're talking about Dungeness crab, that seasonal outlook might actually be more useful. But they're still going to rely on the weekly sampling, weekly testing, all of that will still have to happen. So sometimes I wonder just how useful the model could be when you still need to get down into the public water example to do those regulatory things that fisheries have to do. Not very articulate, but it's one I struggle with because I think in theory it would be nice to have a seasonal forecast, but there aren't too many on the ground strategies that I can think of that one would do if they had that seasonal forecast.
I mean, I guess that's the key.
Yeah.
Information, if they can't reposition their aquaculture to sort of maybe a less at risk place, then maybe have to test
In terms of Zhengchen's crab, the crab pots, it's been hot throughout the seasons up in Humboldt, like hot, toxic-wise in the North Coast, year after year ever since the Pacific Warm anomaly, and so, again, they're not going to make massive decisions to move crab pots around when really you can't quite escape on average that spatial region over which you're going to have HABs, but it's going to happen in fits and spurts along different parts of the coastline throughout the season. So you kind of just got to deal at that granular scale with these things.
Just to follow up on that, I think we just heard that the behavior of the crab fishers was a huge component of the model of the catch, which suggests that they are changing their behavior all the time, or at least I think it does if I interpret that correctly, right?
They're changing at that seasonal scale. You know what I'm saying? Like I think at that seasonal outlook scale, you're not going to see that. You're going to see it on this daily to weekly kind of response.
Following up on that, I guess a question for Annie. I was curious about what aspect of fishers' behavior is part of the model.
Thank you for the question. We include fishing location and date, so day and season, the amount of time that the pots are left, so the soak time. I think that's it, but then we subsample our oceanographic model at those times and locations to match all of the dynamic and lag conditions to those times and locations. So it's kind of embedded throughout our model.
So I had a question in line with what Clarissa was saying, where sometimes we don't know what we need until it's available and we can use it. And regarding these seasonal timescale, would it be possible that there are some socioeconomic repercussions that we're not aware of until these products are available? So for instance, people go and hoard gas when they hear that it might be depleted, right? Or there's lots of reinsurance companies that use climate forecast information. So what if restaurant businesses knew that it would be a bad year for fisheries? Would they drive up the prices and that would help compensate for the fishermen that will have a worse year? Or things like that that we haven't seen those connections, or maybe some people have seen those connections and perhaps could just comment on that.
I can kind of briefly comment on the HAB part. So we don't have data on that sort of thing in California, but there have been a number of studies in Florida who have looked at that human dimensions component, and you do see that economic response to bad Carenia brevis forecasts over a period of time, and you see that in the restaurant industry. So you're saying there's definitely a lot of validity there. And there actually are a lot of things people can do. I was just talking to Sam and thinking about Charlie's question. In California, and this might be true in other parts of the Pacific Northwest, I do know that crab fishermen are often they don't solely fish crab, they have other options, salmon being one of them. And so they do, what I have heard is that if they've invested all their money that year in crab pots, it's hard to then switch to do salmon. But if they knew ahead of time at that seasonal level, maybe they could invest in different gear. So I hadn't thought about that when you asked me, but that's another thing.
I have a question for Antonella. the limb model that you looked at, was it state variable? Did it include atmospheric state as well, or was it just sea surface salinity, sea surface temperature and sea surface cycle?
Was only ocean variables, sea surface temperature and sea surface height at monthly means of these variables. And so sea surface height seems to be very useful. In fact, for this optimal initial condition would seem to be initial condition in sea surface height seem to be really key. I think it's a measure of pollution heat content, so there's some indication of subsurface processes.
I guess I asked because I thought about the marine heat wave as a coupled system where changes in the pollution load lead to an extended high pressure system over an area. And so I guess I'm wondering if would there be a different optimal state if you considered atmospheric variables as well?
So one thing that you can go to, because I wanted to stay in my 5 minutes to give a good example. Once you have your initial conditions, and then you can actually project that those fields on your data and time series, then you can do linear regression of some atmospheric variables. So you have some contours of sea level pressure, which are actually consistent with what we think. So the initial pattern was like an Pacific oscillation pattern, and then once you start having warm anomaly in the tropical Pacific, you develop a little evolution law, which is more consistent with that. So there is definitely that connection between the surface temperature and the atmospheric variables that also leads to this increased persistence depending on what Tom was talking about. While I think the tropics and the connections and the response of the atmosphere to the surface temperature can explain the longer duration, I think the amplitude is most likely due to local atmospheric forcing, which is stochastically tappable in terms of faster time scales. And I think to get into that dimension, we probably need to go higher in temporal resolution than the monthly resolution today.
Hi, I just had 3 questions. I had one first for Ellen. I wanted to hear more about the static, dynamic, and lagged predictor variables. What are those exactly, and what are the differences between those 3? I was super curious about how incorporating that. Like what time scale are the lags at?
Was that for me? I think or Sam. Yeah, so the static conditions are happening at the time and location where the fishing is happening, and those are really focused on conditions that we don't expect to change over time. So like benthic sediment type, latitude and longitude, all the fishing behavior, like the soak time. That's the big one. And then for the dynamic conditions, those are monthly average fields from JScope. And they are subsampled from the model at the times and locations where those plots were set. The lag conditions are subsampled from JScope hindcast, so historical simulations, and those were lagged 3 or 4 years. Does that answer your question?
Yeah, thank you. That's very helpful. I had a question for Marjorie, if no one else's questions. I had a question about the confidence or uncertainty in the mid-century projections. I was really curious about what type of uncertainty were you mainly focusing on? Is it like uncertainty that comes from the data or the methodology, or is there I could also imagine there might be this deeper uncertainty related to maybe the interactions between species that are really difficult to capture, or the evolutionary dynamics at play that might cause uncertainty in their models for your final predictions? I wanted to hear more about that.
Yeah, that's a great question. And so what Kyle really focused on was just three things, and you just mentioned a whole bunch of uncertainty from a whole bunch of different, so there's a lot of uncertainty, but he was really looking, the way he conducted the experiment was to look at the relative uncertainty of which GCM you chose, which downscaling method you chose, and which watershed model you chose. So those were really the three that he was looking at, but you just pointed out that there's lots of other sources of uncertainty. So those were the three that we were looking at, and in fact, and folks always ask, well, what about that emission scenario? Because that's something that is also another certain source of uncertainty in the future. But it turns out by 2050, that's really not significant compared to which GCM you choose. So that didn't play a role. But yeah, so those were the three you looked at, but there's that, it's a good point that we're underestimating the uncertainty if we're only looking at those three, because there's lots of other sources of uncertainty too.
Thank you. And then my last question was just for a lot of people use machine learning methods and designing your models. I was curious about like machine learning models suffer from this problem. Sometimes they can be really difficult to interpret. Right? They can seem like a black box, and they can only focus on it. Sometimes they can only make these correlative conclusions instead of getting at the real cause. And I was wondering if people here had thought about how to get around that. Like, how do you make these systems more interpretable? How can you more directly compare them to mechanistic models to get underlying mechanisms of a lot of the processes that we're looking at? That's my answer. So if people want to talk about this later, that'd be cool.
You can just talk, like, get on that tomorrow. Is he here?
There's probably multiple ways to answer this question, but or if there's an answer, one answer. Ideas, the correlative analysis, I think, can be suggestive of mechanisms and allow you to develop hypotheses to explore further, because these models provide such vast quantities of data that sometimes it's hard to navigate where you should look. And then, the mechanistic side can then be explored in these realistic simulations that are, and then there's the challenge, where is it? Because the reanalysis are really tempting, but in some cases, the act of doing the reanalysis influences the answers you might get, right? So you kind of have to find the right workhorse to investigate that with. And in biogeochemistry, we often rely on budgets for a lot of it, but there's other approaches for that too. And happy to talk more, and I would love to hear what other people think too.
I can add to that a little bit, fleshing it out in ways I didn't get to in my talk because I was going a million miles per hour. But the thing I didn't tell you, like when we were building the dynamic acid model in the early days, we were constraining that around a lot of different, starting with correlations, building GLMs, thinking about really just basic statistics of what's driving dynamic acid in situ. And what kept coming out, and it's come out in a lot of different studies since then in California, is that there are these nutrient ratios that seem to be important. So then we took that and started to build some mechanistic understanding from that. May or may not be right. Time will tell. We might embed it in like a 3D model and see if it stacks up relative to what we know happened. And I think, just building on what Sam said, that's a pretty practical way to think about that leap. And you can do that with machine learning output as well.
One part is that the fun thing about models is that you can also turn pieces off and discover what the role of that is, and that. But that needs to also, there's obviously things to caution yourself with there to use it. That's been a powerful thing for attribution, just kind of what you're getting at,
And that salinity example is a great case. I've seen that happen before, where you're really looking at a process of dilution, vertical mixing, and stratification, but salinity is the proximate variable that shows up. It's not what's mechanistically causing anything.
Yeah, and just speaking about the work that colleagues at that I work closely with, so farming and others are using booster progression trees, even other machine learning techniques in terms of developing species distribution models. And I think somebody talked about it before, but obviously overfitting, problems, so doing cross-validation, and then, but also extrapolation. So, the training data that you have would determine, that's constraining sort of their performance in terms of, let's say, the temperature response over the temperature that they have seen in the training data. But then, if you're using those for projections, for instance, they can give you all sorts of responses that are not really biologically possible, for instance. And if you use different models, they actually will do different things and then extrapolating. So it's important to think about that. And now there are some these shape constraint models that you can constrain the shape of that relationship, even if it's a machine learning model between your SST or other variable and biological output that you're interested in.
Hi everyone. Breakout time. We've got, so I put this list on everybody's table because there were some snafus with the email list this morning, but hopefully you also finally got the email because I had May send it because it clearly didn't like me. But I put a printout of these on each table for all of you in-person folks so you know where to go. And all of the online participants have already been assigned, and we will automatically put you in your breakout rooms. We're going to meet again. It's going to be an hour total, so please spend the first 45 minutes or so in discussion, and then take 10 to 15 minutes at the end to wrap up and create your slide. If anyone has any questions, come talk to me. We're back here at 5:30.
I have no idea, but I think so. Awesome. Yeah, Val will write down. Okay.
It's a very easy one. It's a lot easier than that.
It's hard to keep up with. Yeah, some of us just never leave. So. It's easier, but you guys. So, the thing was, I think the idea here
the in-person fisheries, I don't remember. You go. Okay.
We're staying right here.
You guys fisheries? Okay. We're supposed to be one of your fisheries.
Is that your
Yeah, can I just leave it there because we're coming back.
Yeah, we're coming back.
To facilitate our take notes.
All right. Yeah.